Hello everyone, my name is David Dominic Asokwo from the Department of Biochemistry, University of Uyo. Welcome to our channel. In this video, my team and I will be talking on how to run polyacrylamide gel electrophoresis and AGAROSE gel electrophoresis, the two essential techniques in molecular biology and biochemistry as well. We will also guide you on step by step on how to prepare the gel, loading the sample, and also analyzing the results so that you can understand both the process and the science behind it. Don't forget to like, subscribe, and turn on your notification so that you won't miss any of our future tutorials. We are starting by introducing our first speaker by name Goodness Peter to take us on polyacrylamide gel electrophoresis. Let's get started.
Hello everyone, good morning. My name is Goodness Peter. I will be talking about PAGE, that is the acronym standing for polyacrylamide gel electrophoresis. But before I dive in elaborately on this very aspect, I would like to let us know the meaning of electrophoresis first. Electrophoresis is simply the migration or the mobility or the movement of charged particles, not just particles, through a solution under the influence of an electric field. Some of these biomolecules, such as nucleic acids, nucleotides, peptides, and amino acids, they possesses an ionizable group which help them to exist as electrically charged species in a solution at any given pH. So, and then I want to move directly to PAGE. As I said before, PAGE is an acronym for polyacrylamide gel electrophoresis. And polyacrylamide gel electrophoresis is an important aspect in biochemistry and in molecular biology, even in genetics and in biotechnology. It is used to separate macrobiological molecules based on their size and charge. Polyacrylamide gel electrophoresis has been so much used in the aspect of biochemistry, and it was early introduced in the 1950s by Raymond J. Erickson, and it has helped in protein purification and in protein-to-protein interactions. So we'll be looking at the polyacrylamide gel electrophoresis, how it is used to separate molecules based on their sizes, charge, or conformations. Now, in terms of their sizes, we'll be looking at gel filtration chromatography. Here, it utilizes porous beads, whereby smaller molecules move in first, and then the larger molecules are being excluded, thereby allowing the proteins to be separated based on sizes. And we're going to look at the LDS, which is the sodium dimethyl sulfate. This phase is the phase whereby the protein are being denatured, and then it is coated with the SDS, thereby leaving all the molecules with a negative charge. So as a biochemist, it is expected for you to know this very important aspect in biochemistry. And if you don't know this, you are wrong. And then we are going to move into how they are being separated based on their charge. And under this, we are going to consider the ion exchange chromatography. In the ion exchange chromatography, we are going to talk about how these molecules are being separated based on their net charge at any given pH. Here we consider molecules at different pH, protein-to-protein interactions. For instance, proline coming together with threonine. And we know that we have some essential protein and non-essential protein. So when they come in interaction at different pH, so it is very, very pertinent for a biochemist to understand how this interaction take place. And then we are going to look at the conformations. As I say, the conformation is just simply how these biomolecules are being separated based on shape. And then also you look at their charges as well. Then when we are talking about the polyacrylamide gel electrophoresis, Now let me look at their different conformations. Then we look at polyacrylamide. Polyacrylamide is the gel matrix. Then it is formed by a monomer known as the acrylamide, and then also with the bis-methylene acrylamide, which is known as the cross-linker. So it is used to link these things in together so that we can have a successful protein purification. And then lastly, I will be talking about the gel matrix, the gel, which is the physical medium whereby these samples are being separated. And then also looking at electrophoresis, as I said before, the migration of charged particles like nucleic acid and photosynthesis into different samples, thereby making these samples suitable for purifications. So I am very, very well assured that with these points, biochemists, biotechnologists, they are aware how polyacrylamide gel electrophoresis works. And then I think that with these few points of mine, I've been able to enlighten the whole public on how polyacrylamide gel electrophoresis works. Thank you. Stay tuned. Like our channel, please. View this video and then like and follow our channel. Thank you. As I'm going to hand over to the next person that will be coming to take us more deeply on the aspect of gel electrophoresis.
I'm here to speak on materials used in making the SDS page. Number one, we have the acrylamide and the bisacrylamide. The acrylamide makes the gel, while the bisacrylamide acts like a cross-linker that links the gel together. Number two, we have the APS and the timid. The APS is the aluminum persulfate. The timid is the tetra methyl ethylene diamine. The APS produces the free radicals used in making this gel. It initiates the polymerization of this gel, while the timid acts like a catalyst. It stabilizes the free radicals that makes this gel. Then the third one, we have the trace buffers. The trace buffers has to do with the stacking gel, the resolving gel, or we can call it the separating gel, and the running buffer. The stacking gel is used in, making these proteins go in a straight line to the separating gel. So the pores of this stacking gel has to be big. So to make this stacking gel pores big, the concentration of the acrylamide used in making this gel should be lower, like a 4% acrylamide will be used in making this stacking gel, so that when the proteins go in the stacking gel, it will create like a straight line which passes through to the separating gel. Then the second one, which is the separating gel, the pores has to be very tight because it has to do with separating the proteins. So a higher concentration of acrylamide are used in making this gel. And the pH has to be around 8.8. Then the third one is the running buffer. The running buffer has to contain SDS to keep the proteins denatured. Then the third material we were going to use in making this SDS page is the sodium hydroxyl sulfate, which is the SDS. The SDS acts like a denaturing agent. It's a denaturing agent. It denatures this protein and makes them whole negative. From the positive to the negative, they make the protein lose their shape from the bent shape to a straight shape, so that they'll all be in a straight line. Then number 4 is the sample loading dye. The sample loading dye is being used to make these samples before putting them in the well. They're using making this sample before putting them in the well, and it's used for tracking and monitoring the movement of these proteins so that you can know how far they've gone. And the dye used as a tracking gel, the tracking dye is the homophenol blue. Then the number fourth one is the staining and the staining solution. The staining solution are used after the electrophoresis to visualize the protein bands. So the dye used in making this staining gel, in staining these proteins, are the Kumasi Brilliant Blue or the Kumasi Blue. They are used in staining this gel after electrophoresis. Then the staining gel, after staining this gel, you need to destain it so that you can actually visualize this protein bands clearly, so you can see the proteins clearly. And to make the staining solution, you have to put 10% of acetic acid, 40% of methanol, and 50% of distilled water that is used in making this staining solution. Thank you.
Good day, I'm here to talk about sample preparation, polyacrylamide gel preparation, and also native page. Firstly, I will start with sample preparation. This sample preparation, the goal is to denature all proteins into linear shapes and coat them with SDS, that's sodium geodesial sulfate, to impart a uniform negative charge to mass ratio, ensuring that separation is only based on molecular weight. How to get your sample preparation? That's how to mix it. First, you add in your trice HCl. This trice HCl is the buffer system. Secondly, you add in your SDS, that's sodium geodesial sulfate. It's an anionic detergent that linearizes and coats the proteins. Thirdly, you add in your glycerol. The glycerol work in this is to increase the density of the sample so that it sinks into the wells when loaded. Then fourthly, you add in your beta-mercaptoethanol. This beta-mercaptoethanol is a reducing agent that helps to break the protein's disulfide bond. Fifthly, you add in your bromophenol blue. This bromophenol blue is a small visible dye that moves ahead of the proteins and allows to monitor the run progress. After mixing in the above mentioned samples, that's the sample buffer and the loading buffer, you add them to the protein samples and heat them up for 5 minutes at 95 degrees Celsius in a water bath. The essence of this heating up process is to help to completely unfold the proteins and aids in SDS binding. Then you briefly centrifuge the tube to collect any condensation from the lid and pellet any insoluble particulate matter, because this particulate can clog and cause streaking. Then secondly is the polyacrylamide gel. This polyacrylamide gel is a porous matrix made from cross-linked polyacrylamides that acts as molecular siege. These polyacrylamides are divided into two sections. One, we have the resolving gel, or you can also call it the separation gel. While secondly, we have the stacking gel. The resolving gel has typically 8 to 15% acrylamide, and it also has a smaller pore size with a higher pH of 8.8. This is where the actual size separation occurs. To get your resolving gel, that's how to make your resolving gel. Firstly, you add in your acrylamide or bisacrylamide. Both acrylamide and bisacrylamide are the polymers. Secondly, you add in your trice HCl with a pH of 8.8. Then you add 10% of SDS, that's 10% of sodium geodesial sulfate. You also add in 10% of APS, that's 10% of ammonium persulfate, and lastly you add in your TEMED. This TEMED is tetramethyl-ethylene dianine. Once you add in your TEMED, you mix gently but quickly and make sure it is being used immediately. APS and TEMED is the polymerization catalyst that speeds up the cross-linking reactions. Then we'll also talk about the stacking gel. This stacking gel has typically 4 to 5% acrylamide with a larger pore size and a lower pH of 6.8. It aligns and concentrates all the proteins into tight bands before they enter the resolving gel. To get your stacking gel, like how to prepare your stacking gel, you mix lower concentration of the acrylamide, then you add in your twice HCl at the pH of 6.8. You add 10% of your SDS, that's sodium geodosyl sulfate, then 10% of APS, that's ammonium persulfate, and lastly you add your TEMED, which is tetramethyl dialine. Then after adding them, you mix immediately and use it immediately. Then thirdly, I will talk about native page. This native page is native polyacrylamide gel electrophoresis. This native page is a laboratory techniques used to separate proteins in their natural state. Unlike the SDS page, that's sodium deodeoxyl polyacrylamide gel electrophoresis, the native page does not use denaturing agents like beta-mercaptoethanol. Then the protein migration through the gel depends on 3 factors. Firstly is the size. Large size proteins move slower. Secondly is the shape. Tightly folded proteins moves faster than the loose ones. Then thirdly is the intrinsic charge. The natural electric charge of the proteins in a buffer pH takes the speed and direction of the migration. Applications of this native page are: 1. It is used to study the native structures of proteins. Secondly, it is also used to study the enzymatic activities of proteins or analyze protein to protein complexes. Thanks.
At this stage, we are diving into how to run the polyacrylamide gel electrophoresis. First, we need the electrophoresis apparatus. First thing we need to do is assemble the electrophoresis tank. This includes placing the gel cassette into the chamber, making sure that it is properly sealed. After we have done that, we need to fill the chamber with the appropriate running buffer. The buffer is very essential because it provides ion that carries the electrical current through the gel. After that step, we now load the samples into the well. We take our prepared sample and carefully load them into the wells of the gel using a micro pipette. These steps require steady hand because the wells are small and we want to avoid puncturing the gel or cross-contaminating the wells. Each sample goes into its own designated well. So after we are done with that, we now add the protein ladder. Here, the protein ladder is otherwise called molecular weight marker. So the data contains proteins of known size. It allows us to estimate the molecular weight of the proteins in our sample after the gel run. It serves as a reference or a ruler for interpreting results. After this step, we now load or run it under a voltage. So the voltage setting here is very important for separating the proteins effectively. We begin by running the gel at around 80 volts during the stacking phase. This low voltage ensures that all proteins in each sample line up neatly as they move through the stacking gel. Once they reach the resolving gel, the parts where actual separation occur will increase the voltage to between 120 and 180 volts. Voltage drives the protein through the gel matrix based on their size, with smaller proteins moving faster and farther than the larger. After we are done with that step, we now move on to stain and visualize. The gel that we have already run is very invisible. That's the protein is invisible to the naked eyes. So for us to make it visible, we need to stain and destain it using a proper staining solution and a destaining solution. So after we have stained and destained it, we need to visualize it under a white light box. So when we visualize it under a white light box, we'll be able to see the bands clearly. And this step is very important because it translates all the invisible molecules processes into a clear and visual result that we can interpret and analyze. At this point, I will call on the next presenter, Idara Isaiah, to take us on application of SDS page in biochemistry. Thank you for having me.
Hello everyone, I'm here to speak on application of SDS page in biochemistry. We have determination of protein molecular weight. Under that, SDS coats proteins with a uniform negative charge and unfolds them. This allows the gel to separate proteins only by their size. By comparing an unknown protein band to a known molecular weight marker, researchers can estimate the protein's molecular weight accurately. 2. Assessing protein purity. SDS page is widely used during protein purification process. It is also used as a single band to indicate a highly pure protein sample, also as multiple bands to show that contaminants or other proteins are present. 3. Checking protein expression. In biotechnology and research, SDS page is used to check whether a gene has been successfully expressed in bacteria, yeast, or cell cultures. It helps to further confirm if there is a presence of excess protein, overexpression levels, or correct size of the protein. 4. Monitoring proteins denaturation. Since SDS and heat denature proteins, the techniques helps researchers to understand 1. How stable a protein is, 2. Whether it has completely unfolded, 3. Whether disulfide bonds have been reduced. At this point, I would like to call on the next presenter, Ofonime Augustinede, to take us on the other part, which is AGAROSE gel electrophoresis. Thank you for having me.
Hello everyone, my name is Fonzy and I will be talking on the AGAROSE gel electrophoresis, giving a simple conceptual overview. The agarose gel electrophoresis is a technique that is used to separate high molecular weight nucleic acids such as DNA or RNA based on size using an electric current. Isn't that amazing? In most biological samples, nucleic acids exist as fragments of different lengths, and these techniques enable us to distinguish them. The key idea is that the smaller fragments move differently compared to the larger ones, and this allows us to clearly sort them out. This technique is very important as it helps scientists, molecular biologists, and biochemists to be able to understand the composition and structure of the nucleic acid they are working with. The agarose gel electrophoresis depends on two main things. First is the agarose matrix. The agarose matrix forms a gel which acts like a sieve with tiny pores, and these pores control how fragments move. The second is an electric current. DNA and RNA are negatively charged. When an electric current is induced or applied, all the fragments move toward the positive end through the gel. The combination of the gel and electric current enables those fragments of different sizes to separate from each other. After the separation takes place, the result can be visualized, and this gives us the understanding of what the nucleic acid we are working with looks like. We can also see the size distribution, meaning which fragment is larger or which is smaller. The agarose gel electrophoresis is a technique that is most reliable in the scientific work for separating molecules such as DNA or RNA. At this juncture, I will hand on over to the next presenter as you will be leading us in the next aspect of gel electrophoresis. Thank you for watching.
Hello everyone, I'm Elizabeth Moses ACN. I'll be taking you on the principle of separation of agarose gel electrophoresis. Having been told what agarose gel electrophoresis is, we'll dive down into the separation principle. Principle number 1, charge of nucleic acid. The DNA and the RNA molecules carry a net negative charge due to their phosphate backbone. That is, when an electric field is being applied across a gel, the negatively charged nucleic acid moves from the negative electrode towards the positive electrode. This movement is the base of the separation. Principle number 2, molecular sieving by agarose gel matrix. Agarose gel form a porous mesh-like network when it's solidified. Here, the agarose gel acts as a sieve. Large DNA fragments move slowly because they have difficulty in passing through the small pores. Small DNA fragments move faster because they migrate easily through the matrix. That is, the higher the agarose gel concentration, the smaller the pores, and the better the resolution of the small DNA fragments.
Hi everyone, I'm Blessing Emmanuel, still speaking on how to run AGAROSE gel electrophoresis. Now think of it this way, like setting up a race for molecule. We are going to consider these 6 major components. I'm going to group it into 3.
so that we're able to understand. The first group, we have the arena and the track, consisting of the gel, the buffer, and the chamber. The second group, we have the engine, consisting of the power supply. The third group, we have the runner and the camera, consisting of the sample, the ladder, and the die. Now I'm going to explain them one after another. First on the list, we have our AGAROSE gel matrix. This is our racetrack. Now think of it like a molecular jello or a sponge that the DNA squeezes through. Second, we have the electrophoresis chamber. This is like a tank or a stadium that holds everything together. Third on the list, we have the buffer. The buffer, this is the UV we pour over the gel. It keeps the pH stable and acts as a conductor to allow electricity to flow. The power supply. The gel needs electricity to work. This provides the current that pushes the DNA from the negative side to the positive side. Without this, the runners remain at the starting line. The fifth on our list, that's the DNA and RNA sample. Now, the samples are the competitor running the race. Now, we have the ladder. The ladder helps us measure how far the sample ran. Now, dye or imaging, that's our last part. Now, DNA is actually invisible. So, the dye attaches to the DNA so that when we put it under the light, we'll be able to see the results very well. So, we need gel to separate, electricity to move, and the dye to see the results very well. At this point, I will now call on my colleague Okekere to take us on the conceptual workflow stages of AGAROSE gel electrophoresis. Thank you. Bye.
Good day, everyone. My name is Okekere, and I'll be discussing on the conceptual workflow stages in AGAROSE gel electrophoresis. Agarose is a polysaccharide that is isolated from semen. This polysaccharide is made up of galactose and anhydrogalactose linked together by one 4-glycosidium bond. The agarose gel itself is casted by dissolving the agarose powder into a buffer containing ether and triacetate. The ether then is ethylene diamine tetraacetic acid, and the function of the ether is to bind all the metallic ions present in the buffer, thereby stopping them from interacting during electrophoresis. And this ether serves as a chelating agent, while the TAE itself helps to create a suitable environment for electrophoresis to take place. Then after the mixture of these three components, the mixture is now heated to the point below boiling point so that the agarose powder will dissolve in the solution to form a pure and a clear solution. Okay. Now, when the solution is heated to a point below boiling point, it will form a clear solution. And then this solution on cooling will be poured into a casting tray, which we want and fitted with a Teflon oil cone. And now, after pouring this solution into the casting tray, on solidification, it will form a gel with some pore sizes, and the pore sizes is determined by the percentage of the agarose powder dissolved before heating. And the higher the percentage of the agarose powder dissolved, the smaller the pore sizes, and the smaller the agarose powder dissolved, the higher the pore sizes. Then, after this is being done and solidification takes place, this gel tray is now transferred to the electrophoretic machine, and the electrophoretic machine comprises of the electrophoresis chamber, the power supply, and the electrode. So the electrophoresis chamber is what contains the gel tray, the electrodes, and the buffer itself, while the electrodes is what establishes the current between the gel and the buffer. Then the power supply itself helps to supply voltage in order to facilitate electrophoresis. Now, the gel tray will now be transferred into the electrophoresis tank, and it will be covered with a buffer that was used to prepare it. Then after we move on to sample preparation. At this point, the DNA sample to be resolved will be prepared with a mixture of a viscous buffer. An example of such viscous buffer is 30% glycerol and also a tracking dye. And one of the tracking dye used is bromophenol blue dye. Now, after this, the function of the viscous buffer is to help the DNA sample to penetrate into the wells created by the comb easily without spreading, while the tracking that helps to monitor the migration of the DNA samples from the cathode to the anode. And then note, while placing the gel tray in the electrophoresis chamber, the gel tray is placed at the point that the wells created by the comb is at the cathode, while the opposite side of the wells is at the anode. This is to ensure the migration of the negatively charged DNA particles from the cathode to the anode. Now, after preparing the DNA samples, the DNA samples is now transferred into the wells inside the gel tray, and this is done by the use of pipettes. Now, after pipetting all the DNA samples into the various wells in the gel tray, the gel tank, the gel tank is now covered. And now when the gel tank is covered, at this process, voltage will be introduced into the gel tank by the use of the power supply, which will generate electric current that will enable the transfer of the DNA particles from the cathode to the anode. And once the voltage is introduced, a movement will be noticed. There will be a migration of the DNA particles from the cathode gradually to the anode, and it will be visible by the help of the tracking dye that was mixed in the DNA sample. So once the DNA sample migrates from the cathode to the anode, it is said that the electrophoretic process has been stopped. Then, immediately the electrophoresis process is finished, we now put out the voltage and take out the gel from the electrophoresis chamber. Now, the gel we take out to the electrophoresis chamber will be placed in a buffer containing ephidium bromide and TAE. Now, the function of this ephidium bromide is to help to intercalate the DNA fragment present in that sample. Intercalation is a process whereby this Ethereum bromide will insert itself into the base space of the DNA double strand. So the process is to help make the DNA fragment to be visible when placed under ultraviolet light. What the TAE at this moment helps the Ethereum bromide to intercalate fast as is done, a fluorescent dye will be formed, and this fluorescent dye consists of the DNA to be resolved, or yes, to be viewed. Then, after the fluorescent dye is formed, the fluorescent dye will now be moved or exposed to ultraviolet light for visualization, documentation, quantification, and interpretation. Now, this takes us to the interpretation. So, when the fluorescent dye is exposed to the ultraviolet light, some observation will take place which demands interpretation. Single sharp bands here means that the DNA samples present are identical, and single bands now means the presence of single fragmented genomic DNA. Multiple bands signifies the mixture or heterogeneous sample of DNA, and then smears here indicates DNA degradation or contamination of DNA. And then finally, ladders are used to determine the molecular size of this DNA. Now, I believe that with these few points of mine, I've been able to explain the workflow stages in agarose gel electrophoresis. And now I will call on my fellow teammate by name, Hope Matthew, as she comes to discuss on another topic that is applicable in agarose gel electrophoresis.
My name is Matthew Hopez. I'm here to talk on the applications of AGAROSE gel electrophoresis. It is used for assessing DNA integrity. It helps to determine whether DNA is intact or degraded. Intact DNA will always have flexible band, while degraded DNA shows smearing. It is also used for purity assessment in molecular workflows. You can detect if your DNA sample is contaminated or mixed with other molecules just by observing unexpected bands or smears. It is also used in checking PCR success. PCR means polymerase chain reaction. Now, agarose gel is used to confirm if the target DNA was amplified. Yeah, you will see band as expected time if the PCR works. Also, it is used for analytical restriction design digestion product. When DNA is cut with restriction enzymes, agarose gel helps visualize, that is, it makes it visible for the resulting fragments to confirm successful digestion and check pigment type. Now, to be safety and good laboratory practice, we have to follow the institutional safety guideline. That is, we always adhere to your lab for institutional safety rules to prevent accidents and ensure a safe working environment. We also need to handle stains and bookmarks according to SDL. SDL stands safety data sheet. So it provides safety instructions for chemicals. So we need to follow that to make it suitable. Number 3, we use PPE as supervised lab environment. PPE means personal protective equipment. Yes, like gloves, lab coats, goggles. We wear them to protect ourselves and only work under supervision. If you're a beginner, especially, then you need to ensure electrical equipment is operated safely. Since electrophoresis uses electricity, make sure all wires, power supplies, and tanks are in good condition and used correctly to avoid shocks and fire hazards.
In conclusion, we hope that this video has helped you understand how agarose gel electrophoresis and the polyacrylamide gel electrophoresis are applied in biochemistry in separating molecules. This technique is a very vital tool in research and molecular biology as it deepens our understanding of separation in the scientific world. Thank you for watching. Do well to like, comment, and subscribe to our channel for more educative content. Thank you.